Okay, welcome back, everyone. Our next speaker, it's my pleasure to introduce, Dr. Andrea Brunel. Andrea is an associate professor in the geography department at the University of Utah. She's a paleoecologist who uses lake and wetland sediments to study past environments with a focus on disturbance and issues related to land management. Andrea has a PhD in geography from the University of Oregon in Eugene.
So I'd just like to start by thanking the organizers and Darren in particular for inviting me. It's a real honor to speak to this group and at this conference. I also wanted to mention, I was really excited to hear about the objectives of this meeting, to not necessarily talk about things that I've done, but things that need to be done and as a group that we need to do. So when I had to come up with my title, it was an introduction to paleoecological data and their utility in restoration or ecosystem restoration. But maybe a better title might be, what can we do in the future with data from the past? And I appreciate having the Julio and Connie go ahead of me, although they set kind of a high bar for the talk, because they provided some context for these long-term changes that paleoecological records can present. What I'm going to be doing today is talking about paleoecological records in general. What are they? How do we get them? What types of data do we get? What do they look like? And what I've been trying to think about these last few years is how can we use them better? How can we actually use them in a way that might facilitate land management or restoration or adaptation, or however you want to put it, the things that we're trying to do to ensure that we have ecosystem health into the future. So the idea behind paleoecological records is, in my case, I work on sediments, and to do that you need to have a sedimentary basin, so a basin where sediments and proxy records are accumulating over time. These can be lakes, and in many cases they are, but they don't have to be. They can be bogs, wetlands, wet meadows, marshes. Any place where sediments are accumulating, we can work. And preferably, these sediments stay wetted because that's best for the macrofossil and pollen preservation. And of course, the idea is that things like pollen that's blowing around on the landscape from the plants, charcoal that's being released from fires, and if we're really lucky, insect remains, get captured in these sedimentary systems. And as the systems produce organic sediment over time and the layers accumulate, we end up with a stratigraphy of these vegetation communities and disturbance regimes, with, of course, the oldest sediments at the bottom and the youngest sediments at the top. Oftentimes, these lovely sedimentological records are covered by some meters of water, so we have to go out and get them, which can be the funnest and most challenging part of the whole business sometimes. We use an anchored platform in the middle of a lake, and from there we use a device called a Livingston Core, which is essentially just a meter-long steel tube that has a piston in it. And so you can see the piston loose here. Usually it's inside the tube and it slides up and down as it's attached to a cable. And the whole principle is based on suction, sort of like if you stick your drinking straw in your milkshake or your soda or whatever, and you put the thumb over the end and you can lift it up and the liquid stays in the straw. The same idea applies here, where we push it down into the mud. As we're pushing the tube into the mud, the piston slides up the tube, so that when we pull it out, the mud doesn't just get sucked back out. We can bring it back up onto the boat. Here's a group of my graduate students out coring in the Bear River Range. It's lots of fun, you can tell. And this is what the cores look like. They're 2-inch diameter cores. We bring them back to the lab. We describe the color, changes in grain size, and then we sample them, cut them, and we keep them under refrigerated conditions to discourage mold and things from growing on them. And from within these samples, these slices of the core, we look at a variety of different proxy, so indirect measures of the past environment. And these things include pollen, plant macrofossils, charcoal, and, as I mentioned, insect remains. So I'll talk about insects first. Probably most of you in this room know more about bark beetles than I do, so I'll just introduce them briefly. Basically, they burrow into the bark of the trees, creating egg galleries. You can see eggs and larvae in this photograph. The evidence on the tree are these resin tubes. And the beetles that I'm particularly interested in are the spruce beetle and the mountain pine beetle. We know that these insects are native on the landscape. I have a couple records from the northern Rockies of Idaho and Montana that have records of mountain pine beetle from about 8,000 years ago. They can occur in small endemic scale outbreaks, just a little bit of low amounts of tree mortality. Or we can see things like this. This is a picture from the Wasatch Plateau. Some of you may have been up there and seen the high levels of spruce mortality from an outbreak in the 80s that we've had. This is the Blue Lake Basin, and all this standing dead is Engelmann spruce. The live trees that you can sort of see in between are mainly fir that are coming up from the release of losing all that spruce. So what do these data look like? These are those records I mentioned from the northern Rockies. I have Hoodoo Lake in Idaho and Baker Lake in Montana. And paleoecologists, core people, tend to always present their records with time on the y-axis. I know for a lot of people that's not necessarily intuitive, but if you think about it the way we do, the mud is in the ground and top is at the present, is at the top, and some thousands of years ago at the bottom. So that's what we have here. Present is at the top. And we have 12,000 and about 14,500 years ago on the bottom, respectively. And the column to look at is this one right here that says MPB. And you can see that in Hoodoo Lake, we had 2 occurrences of mountain pine beetle around 8,000 years ago. And then in Baker Lake, we had evidence. We had an elytra, or a couple, from the 1920s, 30s outbreak in Montana. And then I believe there were 4 separate samples around 8,000 years ago that had the mountain pine beetles in the record. And I'm going to come back to this diagram in a minute to talk about what some of these other things are. The next thing I want to talk about is pollen and plant macrofossils. Pollen is probably one of the better known, more common paleoecological records used because it's so widespread in the landscape. Pollen is produced and then distributed across the landscape, and in continually wetted conditions, it preserves really well. You can get pollen that's just thousands and thousands of years old. And the idea behind looking at these pollen grains and plant macrofossils is that the pollen that you find in a given sediment sample represents the vegetation that was on the landscape at that time. So the example I show here is a change from a tree-line environment, something you might see post-glacial. So as the glaciers retreat, you have kind of an open tree-line, parkland type environment. And then as climate warms after the glaciers receded, you see the development maybe of a pine forest. And we can identify this really well in those sedimentary records by the shift in the different types of pollen that are present. Now, pollen in many cases can't be identified to species. There are a few cases where we can, and even a few instances where we can get to variety. But we can tell family and get to genus, and so we can tell these community shifts, these changes in communities of plants across the landscape. And since plants have climate space that they tend to occur in, we can then infer what the climate change was associated with those vegetation changes. We also look for plant macrofossils. Here we have a larch needle. Pollen is, in some cases, designed to be spread far across the landscape. Here we have a pine grain, and these 2 little Mickey Mouse ears on it are actually air bladders. The pine pollen is designed to go long distances. So while you get a representation of the local signal, you can actually be getting pollen from other places outside your watershed. Plant macrofossils don't travel as well. If they get into a stream, they tend to get degraded. So if we find a cone, a male cone, female cone bracts, pine needles, spruce needles, we know that those species were in pretty close proximity to our site, and that can help us better interpret the pollen record. And this is what a pollen diagram looks like. Many of you have probably seen these and then quickly turn the page. Believe it or not, we make these with the DOS program still. It's a specialized software for pollen geeks. But they're not as bad as you think. So again, present at the top, past at the bottom. And each panel represents a different plant taxa. So the samples run horizontally across the diagram, and then the different taxa that we find in each sample are the vertical axes. So for example, this first panel is sagebrush, and what you can see is that there was a lot of sagebrush early in the record. And it's probably not big Great Basin sagebrush. This is probably a high elevation herbaceous type, because it's occurring with spruce and grass and herbs. And then what you can see here about 14,000 years ago is we see a shift from this open parkland, probably above tree line type environment, to the pine taking over. It takes 1,000 years or so for the pine forest to develop, as evidenced by 80% of the count being dominated by pine. A shift in the middle of the record to a pine and Douglas fir forest. We see Douglas fir, which is right here, becoming more abundant. We also see an increase in alder, and then the modern forest developing around 2,500 years ago or so. You can look at pollen diagrams like this to present these types of data. We can summarize them in these panels that just look at the big changes in vegetation over time. And going back to this, we can actually look at specific taxa, ratios, relationships between different taxa. Because the idea is to not necessarily just use a blunt instrument. We want to actually ask specific questions of these data. And in this case, I was looking at these mountain pine beetle instances and trying to figure out what was the dominant pine species on the landscape at the time of the outbreaks. So what I did was I took a ratio of the lodgepole pine type to the whitebark pine type and created an index. So this is something that we can do with these data. You can take two taxa that represent opposite ends of a spectrum, or summer wet versus summer dry, winter wet versus winter dry, cold versus warm, and create an index to look at the change in that parameter over time. And so that's what I've done here. That's one thing we can do. Another way to look at pollen data is to actually use pollen influx. So in the previous figure, we're looking at percentages. So if one of the taxa increases a lot, then everything else has to decrease proportionally because they're all based on percentages. This is a really good way of getting a general view of how the vegetation changes over time, but it can be misleading as far as actual amounts of pollen coming into the system or actual abundances of plants on the landscape. And so one way to get around that is to look at this pollen influx. And if you look over on the right diagram, the Baker Lake diagram, you can see that by looking at pollen influx, which is actually providing a measure of the grains per centimeter squared landing on the surface of the lake per year, you can see that Abies is actually a lot more abundant following this occurrence of mountain pine beetle. But if we look back at the percentage diagram, and here's Abies right here, the fir, you don't really pick that out. So these are things that you might not necessarily notice if all you looked at was percentage alone. So there's a lot of information that we can get from pollen that isn't necessarily presented in a standard diagram. So what I'm trying to think about is how can we use these data to be more applicable to management questions. So pollen, insects, last but not least, charcoal. Get charcoal when there's fires. It's time consuming to count. We count samples from every single linear centimeter of the core. So if we have a 3.5 meter long core, we count 350 samples from that core. And basically we just get a petri dish with a volumetric sample and count how many particles we find. Not rocket science. It's black, it's shiny, it's opaque. It has cell structure, but it is time consuming. And even though there's great joy in counting thousands and thousands of pieces of charcoal, the ultimate goal, yeah, you guys are all lining up to come and work in my lab, I bet. The ultimate goal is to get to something that gives us a representation of fire history, right? How is fire regime changing? How have they changed in the past? So we go from concentrations, just a representation of those counts, and we convert it to influx. Again, we're interested in the relationship of time in these proxy. So that's what we're looking at here in this second panel. Influx, which is the black line, and then background, which is actually a smoothed weighted mean that we run over this influx. And the idea behind background is the amount of charcoal coming in a basin can change for reasons other than a recent fire. Certainly, charcoal can be stored in the sediments. following a fire, and then if you have a big erosion event 20 years later, those charcoal will be pushed into the lake basin. If you have a big shift in vegetation, you go from maybe shrub steppe to forested system, your fuel load is going to change, and that's going to change the amount of charcoal coming in the system. So we try to account for that by looking at background. And then what we want to identify are peaks. What are the fire events in the episode, or excuse me, in the system? So when the influx exceeds the background, so when the black line exceeds the red line, we have it register as a peak, and then these peaks are smoothed over time to give us a fire frequency. Classically, these records have been presented as episodes per 1,000 years. So how many fire episodes per 1,000 years? Now we use episodes because we're trying to be conservative. I think it was Peter yesterday pointed out that paleo records are great for length, but resolution isn't necessarily so great. And this is true. We often have 10 to 20 years represented in each centimeter that we're looking at. And while in subalpine systems it's unlikely that we had more than one fire in a 10-year period, it's possible. So instead of saying fires, we say episodes, just to be conservative. One way that I've been thinking that we might make this data more accessible and eliminate a step of thinking is we're saying episodes per 1,000 years. It might be more useful as a fire return interval. So instead of saying how many fires per 1,000 years, what's the fire return interval? So this would be a fire every 250 years instead of 4 fires every 1,000 years, which might make it more intuitive. So what I'd like to talk about next are just a couple examples of some research that I've done where I've taken paleoecological data and worked with, or am working with, land managers to try to come up with information that can be used. One of the sites or studies is close to home here in the Great Basin. It's looking at the spruce beetle outbreaks on the high plateaus in Utah. As many of you know, there's some debate about what the role of spruce beetles is in these systems. Should there be fires coming through following an outbreak? Should we leave the standing dead? What's the successional pattern? And certainly Tom Veblen and his group in Colorado have contributed to the understanding of spruce beetle outbreaks in these subalpine systems. However, as wonderful as Dendro is, it doesn't go back that far. So what we're hoping to do with these pollen records from these high plateau sites is to provide an even longer record of the return interval of epidemic spruce outbreaks, to look at over long time scales, over 15,000 years, do we see any relationship between when we see a spruce beetle outbreak on the landscape and fire? What's the vegetation response on these longer time scales, going back before human settlement by thousands of years? What can we learn from this? This work is ongoing, so we don't have the answers yet, but right now it's been really interesting. The fire records don't look like fire is a big component to these systems, like Veblen's work has indicated. We're seeing few and not very many large fires over long time periods. And just another shot of Blue Lake. Another project a little bit further away, but I think still relevant to the Great Basin, is some work that I've been doing on desert wetlands. These desert wetlands, or cienagas, are very interesting systems. They're refuges for waterfowl and fish and amphibians and a few listed and endangered plants. And one of the sites that we're working on is right on the Arizona-Mexico border, just east of Douglas, Arizona. It's called the San Bernardino National Wildlife Refuge. Now, a lot of these desert wetlands are no longer functioning. The water table has been dropped so far that in order to get water to the surface to provide the resource for the animals and the plants, they've actually had to sink wells and are pumping and filling ponds. As part of the restoration effort, the San Bernardino is actually working with a private ranch on the Mexican side, Rancho de San Bernardino, and the folks down here are actually aggressively trying to restore and lift the water table back up. They're putting in gabions, or these chicken wire rock-filled check dams, to try to slow down the flood pulses and encourage sedimentation. And they're actually having some quite good luck. They've actually restored, and I'm leery of using that word now, they've brought the function back to a cienega, where the water tables come back to the surface, the aquatic vegetation has come back, there's sedges, it's being used by migrating waterfowl. The question that they wanted us to answer is, what was there before? Was fire a part of these systems over the long-term record? Because as they're getting the water table back up and the systems are starting to function, they're not sure what to do next. So what we did was a pollen charcoal, kind of our bag of tricks reconstruction. And it actually ended up being a really, really interesting record. I'm just going to show you part of it. Again, time on the y-axis present at the top. We're looking at about 8,000 years. And this first panel is charcoal accumulation rate. So again, this influx, the particles per centimeter squared per year. And I have to admit, we were getting nervous because we started at the bottom and working our way up, and there was no charcoal in the sediments. None, except for one piece here, until we got up to about this depths that were about 5,000 years old. After this point, we found abundant charcoal, and in the period of time associated with the medieval climate anomaly, even more. And like Julio mentioned, we were actually seeing stepwise changes in the fire history. So going from nothing to significantly more to even more than that. And what's interesting is this record corresponds quite well with the onset of ENSO conditions. And right around the time that we have other evidence of ENSO circulation initiating, that's when we start seeing fires on these wetlands in the desert. And again, to tie it into what Julio said, there's a disconnect between how El Nino or La Nina affects fires in the desert versus how they affect them in the forest in the southwest. What I think is interesting is it seems that climatic variability is what's allowing, it's the wet and drying cycles that's what's allowing fire to occur on the wetland. And it's been really interesting because it shows that there have been fires over time. So I just wanted to wrap up there so we'd have time for questions. I'm, as I mentioned, really interested in hearing what you think. Are there ways that these types of data can be presented better? Questions that I'm not thinking to ask. I really like working with Forest Service and BLM and Fish and Wildlife scientists and land managers. It feels like it's a really good partnership. So that's it. Yes.
What did you say to them? I mean, what did they take away from what you did?
They were very interested because they were actually, before we did the work, they were doing quite a bit of fire suppression with the ignitions that were occurring on the wetland. And so I haven't talked to them lately, but the last I heard, they were actually considering letting some of the fires burn across the wetland surface. Because, during the drier parts of the year, the tops of the aquatic vegetation will dry out, and the fires actually just burn right across that, and then it deposits a bunch of charcoal. And so last I heard at Los Ojitos, the one that they'd restored a little further south, they were planning to let the next ignition go.
In your record from sediments, how well are you able to piece together fairly complete pictures of plant communities, or are you getting sort of representative bits and pieces like, dominant conifer species or shrubs, etc.? Are you able to sort of tease out if there's changes in association, perhaps with herbaceous species?
Definitely. Oh, sorry. The question was, with the pollen records, are we able to just see big changes in the dominant taxa over time, or can we tease out changes in the herbaceous taxa? Is that correct? We can definitely get to that level, and the way that we've been doing that is by using those influx data that I mentioned. So instead of just looking at broad changes in percentages, we actually look at changes in the herbaceous component, grasses, shrubs, and pull their influx out independently and see how those abundances have changed over time. And we've actually been able to see some really interesting post-fire response. So the non-arboreal taxa increased pretty significantly following a fire. And those are actually, that's the way we're at high resolution sampling, looking at the pollen vegetation response to the beetle outbreaks as well. So we're having quite good luck with that.
I'm sorry, I'm a member of the public and not very well informed about all of this. But when you said in Mexico they had found ways to raise the groundwater, not to the original level, does this have any application in restoring aquifers? We were hearing about that yesterday. Perhaps insoluble problems.
You know, I think that's a good question and may go beyond my... Oh, the question was the work that's been done in Mexico to raise the water table and get the water table back at the surface of the cienagas are wetted. Is that having application to bringing the aquifer level back up? In those situations, it's a really local response. So we have perched systems anyway, and basically what's happening is instead of allowing the stream to keep cutting down and bringing the water table lower down to its base, it's allowing the sediments to accumulate back up so the water table rises back up. I think the problem you're talking about is more a recharge problem where there's more water being taken out that's being replenished back into the system. So I think they're two different issues.
To what extent does spatial variability in the sediments hinder your abilities to get accurate information?
I'm not sure I understand your question.
So you have a lake and you have to pick a core. You take a single core, you take 10, you take 20, or you just rely on that one core?
That's a great question. So he asked, when we take a sediment core, do we take 1, do we take 2, do we take 20? How do we know that what we're collecting represents the basin? That's a really good question, and one that's actually kind of plagued paleoecology for quite a while. There's a few studies that have looked at multiple cores within a basin, and when we started, it's something I've always wondered about too. So when we started this Wasatch Plateau research, we actually took 26 cores from one lake, because usually you take one, because by the time you look at the pollen and the charcoal and everything from a single core, there's 2 years of your life. Seriously. So to do that over, repeatedly, so what we did is we took 26 short cores and we looked at the charcoal, the macrofossils, the loss on ignition, which is an indicator of productivity, the magnetic susceptibility. And what we found is that they agreed across the basin. So we felt comfortable by choosing then a single site and a single core to work up for the history.
Do you think your data would be useful to look at the range of the precipitation or range of beetle parts and extent outbreaks to calibrate the density of pieces you see in the board to say that, I get 5 elytra per square centimeter that corresponds to an outbreak on 500.
Yeah, that's a great question. So can we use the modern outbreaks in these basins to calibrate, like what does the spatial scale of the outbreak, how does that get represented in the basin? The elytra have been challenging. I happened upon them when I was working in the Northern Rockies and just assumed that any place there'd been a beetle outbreak, we would find them. We're actually finding out that and we're doing a lot of calibration work on this question because they're just not as easy to find as we thought they would be. So we're looking at basins that have had historic mountain pine beetles, historic spruce beetles. We're looking at not only the primary beetle, but also secondary beetles, because there's a suite of secondary beetles that can come in following an outbreak, so we're looking at them. And then we're looking at every single sample for pollen, which you may or may not know, but that's hardly ever done. Usually you take a pollen sample every 500 years of your record and call that good. We're looking at every single sample, so we're looking at very, very high resolution pollen reconstructions, and not just relying on having the beetle parts. It's looking at the whole response of the basin. What does sedimentologically, pollen, charcoal, organics, magnetic susceptibility, erosion, what does a beetle outbreak look like in the sediments? So that if we don't have the beetle parts, we know what it looks like. But we're also looking at students working with Barb and I up in the Uintas right now, looking at modern beetle outbreaks and the abundances and secondary beetles to try to get at that as well.